Thank you for standing by and welcome to the Paradigm Biopharmaceuticals Topline Data Investor Webinar, discussing the announcement lodged with the ASX earlier this morning. Firstly, an apology for the short notice between the announcement lodgement and this session. We were awaiting approval from the ASX to have the announcement lodged. And for anyone who has missed the live session now, this is being recorded and the replay will be available very soon after the end of the webinar. All participants today are in a listen-only mode. There will be a slide presentation lasting for approximately 20 to 25 minutes. Please note that today's session is designed to give an overview of the new data announcement, and we would ask for any questions to be referred to the investorrelations@paradigmbiopharma.com email address, which can be found at the bottom of all announcements, and these will be responded to after the webinar. In terms of introductions, appearing on behalf of Paradigm today, we have CEO Marco Polizzi, Chief Medical Officer Dr. Donna Skerrett, and Chief Scientific Officer Dr. Ravi Krishnan, and Director of Investor Relations Simon White. To begin, I'll hand it over to Marco. Please go ahead.
Thank you, Matt, and thank you everyone for joining. I am Marco Polizzi, the CEO of Paradigm, and as Matt just introduced, we'll have our Chief Medical Officer, Dr. Donna Skerrett, our Chief Scientific Officer, Dr. Ravi Krishnan, and Simon White, Investor Relations, on the call with me today. But let me start by saying the top-line results reported today from this phase 2 clinical trial is a fantastic outcome for Paradigm and our shareholders. Not only have we been able to meet our primary endpoints, our top-line results of change in baseline at day 56 in one or more synovial fluid biomarkers, and have observed favorable changes compared to placebo in 6 biomarkers associated with pain, inflammation, and cartilage degradation. But we've also achieved statistically significant improvements in WOMAC, pain, function, and stiffness scores in the twice-weekly PPS group compared to placebo. And I'm excited for the team to present today the results to you. Dr. Skerrett will first off remind investors why this phase 2 clinical trial was undertaken and detail the top-line results observed at day 56. Dr. Krishnan will provide an overview of the top-line data in the complementary study of PPS in osteoarthritic dogs. And then Dr. Skerrett will discuss the next significant time points in this program, the 6-month follow-up, and what Paradigm looks forward to presenting to shareholders in the first quarter of next year. But this is really encouraging data, and it'll be very valuable in our discussions that we intend to have with key regulatory bodies and potential commercial partners. And with that, now I'd like to turn it over to Dr. Skerrett.
Thank you very much, Marco. So why did we conduct this study? Well, you may know that previously we conducted a phase 2b study, PARA-005, where we evaluated the effects of PPS compared to placebo control. And we looked at pain and function, but we also looked at some endpoints, such as MRI to evaluate bone marrow lesions and reduction serum markers, such as comp Adams TS5 and CTX2. Now, on the left side of this slide, you see some images showing reduction in the size of bone marrow lesions. In this patient, you see that the lesion was grade 3 at the time of baseline, and then following PPS treatment at day 53, this lesion had regressed to grade 2. And there is important literature indicating that BML is not only an indicator of increased pain in osteoarthritis, but also the severity of the disease and the progression to requiring knee replacement. So we know that BML lesions are a marker for more severe osteoarthritis, and we're showing here that PPS demonstrated reduction in that marker of severity and progression. On the right, what we're looking at are some known markers of cartilage degradation. So in the serum of patients treated with PPS, as you see in red, after the treatment, they decreased the amount of these markers of degradation in their serum. We saw that for COMP in the serum, we saw them for Adams TS5 in the serum, In the case of CTX2, it's measured in the urine, and so we saw that decrease in the urine. And in contrast, you see in the placebo controls, those markers increase. So those increases are telling us that the disease progressed in the placebo controls and improved in the IPPS treated subjects. So this is evidence in the bone marrow, evidence in the serum. Our next question is, so what about the joint? What's actually going on locally in the index knee that's causing the pain? And to evaluate what's going on in the index knee, we conducted the current study, Para-008, which we'll tell you about more today. But the objective of that study was really to understand what is going on locally in the knee with regards to biomarkers. Next slide, please. So why do we care so much about biomarkers in the serum, urine, or in the synovial fluid? These are important clues to the mechanisms of action of PPS in a certain disease state. We know that PPS has multiple modes of action. They are effective through NF-kappa B, as you see on this diagram, and they also have direct effects. So through NF-kappa B and directly, PPS has effects on decreasing inflammation, decreasing pain, tissue preservation, and improvement of blood flow. And what we've acquired with the data from this study, just the synovial fluid to date, is that we're seeing evidence of some of these mechanisms of action through measurement of biomarkers in the synovial fluid. So we have added to our understanding of mechanism of action in the areas of effects on inflammation, pain, as well as tissue preservation by conducting this study. Next slide, please. So here's an overview of the Para-008 phase 2 clinical trial. It was conducted, as I said, to evaluate the treatment effects of injectable PPS on synovial fluid biomarkers associated with OA-related pain, inflammation, and disease progression. 61 patients participated, were randomized 1:1:1 to receive injections of PPS either twice weekly, once weekly, or a placebo arm. The treatment course was 6 weeks. This trial was run at 2 sites in Australia, and they aimed to gather data on the medium to long-term structure modifying and symptom modifying effects of injectable PPS on the OA. The primary endpoint is the change from baseline at day 56 in one or more synovial fluid biomarkers associated with disease progression in OA. The phase 2 trial also is assessing a number of key secondary and exploratory endpoints at various time points out to 1 year. And those include WOMAC pain and function data, MRI changes in the bone and the joint, and then correlations between synovial fluid changes and clinical outcomes, including pain and function. Next slide. So what have we seen? As we've heard from Marco, we're very excited about the day 56 top-line results, which have shown us changes in synovial fluid biomarkers. This adds a tremendous amount of information to our understanding of how PPS works in the synovial fluid, as well as and consistently with prior data that we've seen in the serum. IPPS impacted multiple biomarkers measured in the synovial fluid. NGF, nerve growth factor reduction, was shown and indicates a mechanism relating to pain reduction. Reductions in TNF-alpha and IL-6 indicate mechanistic effects on inflammatory pathways. And reductions in COMT and human Rs and increases in TIMP1 are providing us important insights on how injectable PPS mechanisms impact cartilage preservation. Important to remember is that in all of these cases, the synovial biomarker changes that we observed in the IPPS treated subjects at day 56 were favorable compared to the placebo control. And so that means the changes that we've show that PPS effects are favorable on the patient's biomarker profile compared to the placebo control responses. Next slide. Now, we also wanted to look at pain and function in this study. We have a lot of experience with WOMAC pain and function endpoints in PPS, and it was important to capture that in this clinical trial as well. So our participants were asked to provide baseline scores using the WOMAC osteoarthritis index. IPPS treated patients showed statistically significant improvements at day 56 in pain, function, stiffness, and the overall WOMAC score for the twice-weekly IPPS arm compared to placebo. The mean percentage change from baseline in WOMAC is 50% compared to 30% placebo, with a P of probability equal to 0.05 for twice-weekly IPPS compared to placebo. And for the mean percentage change from baseline in WOMAC function is 50% compared to 25% for IPPS compared to placebo. Again, the twice-weekly group with a p-score of less than 0.05, p being 0.017. So both of these p-scores are considered significant statistically. The proportion of patients achieving greater than 30% and greater than 50% improvement in their pain overall were 73% and 60%. So that you see here that the vast majority of patients reported clinically meaningful improvement in their WOMAC pain over the course of treatment. Reduction in pain for IPPS treated subjects is consistent with the clinical effects that we've observed in this and prior studies of PPS and osteoarthritis. Next slide, please. I'll now hand the discussion over to Dr. Ravi Krishnan to share the observations from the OA Canine model, which are also adding important information to our program of osteoarthritis.
Thank you, Donna. In this slide, I'll be discussing the canine model. Paradigm has been conducting a trial in dogs with naturally occurring osteoarthritis to support the in vivo mechanisms of action of PPS for potential disease modification and to provide complementary data to the PARA-008 human clinical trial. Since both in humans and in canines, osteoarthritis is a progressive degenerative disease and is influenced by similar risk factors, the canine study of naturally occurring osteoarthritis is a highly relevant translational model compared to rodent models of osteoarthritis. Furthermore, the dog's lifespan being shorter relative to that of humans is potentially advantageous in rapidly evaluating the ability of injectable PPS to modify OA disease progression in a shorter time frame to analyze OA joint structural changes, considering, for example, 20 weeks in a dog's lifespan is equivalent to a 3-year period in humans. The canine study is ongoing and consists of 21 client-owned dogs of varying breeds that are presented at the University of Veterinary Werribee Animal Hospital in Victoria, in Australia, and the dogs were assessed for lameness. Dogs of both genders with either radiologically and or clinically defined osteoarthritis of the hind limb and the front limb are progressively screened and randomized into 2-to-1 ratio to either treatment with subcutaneous PPS at a dose of 1.7 milligram per kilogram human equivalent or saline, which is a placebo group, weekly for 6 weeks, respectively. The key data that's sought from this study are changes from baseline compared to week 8, which is equivalent to day 56, and week 26, which is equivalent to 3 years in human terms. And essentially, what we're looking at is joint function, which is assessed by percentage body weight distribution in the affected limb and is measured by a total pressure index percentage. And also we're looking at biomarkers of joint degeneration within the synovial fluid and in serum, structural changes were also determined by osteoarthritis clinical scores as assessed by X-ray and MRI. Have the next slide, please. In the next slide, we report early interim functional data in 9 osteoarthritic dogs treated with injectable PPS. Joint function is assessed by percentage body weight distribution in the affected limb and is measured by the total pressure index, TPI percent. So improvement in the TPI percent scores is defined as a numerical increase in the value of the affected limb. That's what we term improvement. So early top line results indicate that 7 out of 9 osteoarthritic dogs treated with injectable PPS demonstrated a clinically meaningful functional improvement in TPI percent in the affected limb when assessed at 8 weeks. So this is 2 weeks following the final injection of PPS compared to baseline. In 4 IPPS treated dogs, the front limb demonstrated a mean percentage change, which is an improvement from baseline of 5.6%, and similarly, a positive trend of 10% improvement in TPI was observed for the hind limb of 5 PPS treated dogs at 8 weeks compared to baseline. A mean increase of 5% in TPI is deemed clinically meaningful improvement. So the canine results here provide supporting evidence of functional improvements following PPS, which parallels the observations in the clinical study reported in Para 008. I have the next slide, please. In this slide, I will report on our early biomarker observations in osteoarthritic dogs following PPS treatment at week 8 compared to baseline. PPS treatment in osteoarthritic dogs may demonstrate a reduction in cartilage degradation biomarkers locally within the synovial joint and systemically via serum analysis. We have defined responders here as that those that have demonstrated a reduction in the level of a specific biomarker at week 8 when compared to the biomarker level at baseline. At the time of analysis, synovial fluid biomarker data was only available for 4 out of the 9 dogs compared to serum samples. So in summary, hyaluronic acid and aggrecan degradation neuroepitope. In the dog, it's ARG, which is equivalent to the human ARGS, are cartilage degradation biomarkers. And what we have seen is 3 out of 4 dogs had reduced levels of aggrecan degradation. 4 out of 4 dogs had reduced levels of hyaluronic acid in the synovial fluid. Analysis of serum biomarkers demonstrated that 3 out of 6 dogs showed a reduction in serum ARG, and 5 out of 9 dogs had reduced serum hyaluronic acid, supporting the effects of PPS that we have also shown in the synovial fluid. Reduction in ARG levels within the synovial joint supports the in vivo mechanism of action, since PPS inhibits ADAMTS5 enzyme, which degrades aggrecan in cartilage to produce ARG. Degrading cartilage matrix releases hyaluronic acid into the synovial fluid in OA, and this is reduced by PPS as shown in this study. Recruitment for the canine OA model remains ongoing to complete treatment groups to enable meaningful comparisons to be made with the placebo group. The longer follow-up period at week 26, equivalent to 3 years in human terms, will allow for collective analysis of pain, function, joint structure, biomarker levels following PPS treatment, and will provide informative data to assess the potential of PPS to inhibit the progression of disease as measured by structural outcomes that will define its activity as a D mode. While these early preliminary data demonstrate a trend towards reduction in cartilage degradation biomarkers locally within the joint, in the synovial fluid, and systemically in the serum. The complete study report examining both week 8 and week 26 responses in the final cohort of dogs will be reported in the first half of calendar year 2023. So I will now forward to Donna to present the next slide. Thank you, Donna.
Thank you, Ravi. So what's next? What we expect over the next 6 months. For this study, Para-008, we will continue to monitor patients and report out on the durability of effect in clinical outcomes, WOMAC pain, function, stiffness, overall, and PGIC. We will look for changes and correlations between the synovial fluid biomarkers, serum, and urine biomarkers, and look for correlations with changes in these biomarkers with clinical outcomes. We also have at the 6-month time point MRI changes in the bone and joint, and those, of course, will be correlated with clinical and biomarker findings. All these data in aggregate will help us put together the disease-modifying potential of this drug in osteoarthritis of the knee. Importantly, putting together a disease-modifying pathway based on mechanistic data allow us to have discussions with regulatory authorities around the regulatory pathways for disease modification. And those are important to us to understand what the regulators would need to see in clinical data in order for us to extend the data to best reflect the effects of PPS on OA of the knee. In our canine study, we're very eager to see the end of the study because it gives us a real snapshot of what to expect with 3-year human data. So that study also is ongoing. We'll see durability of effect of joint function, biomarker changes within the synovial fluid and in the serum over time, and structural changes will be assessed by OA clinical scores and by X-ray and MRI. So as you can see, we have this combined data sets of both human data as well as canine data. They're covering a broad range of outcomes, including mechanistic measures, clinical measures, and radiographic measures. And they're really starting to point us to the direction of understanding what the mechanistic effects of PPS are in the knee and the serum, and how these will put together the story that we believe is pointing us in the direction of cartilage preservation based on the signals we've seen from these 2 studies. So with that in mind, I'd like to hand the conversation over to Marco to provide a broader view of what to expect over the next several months. Thank you.
Great. Thank you, Donna. Again, I want to close by reiterating how pleased we are with these results. This phase 2 study demonstrated changes not in only 1, but in 6 key biomarkers compared to placebo. And it also demonstrated consistent, statistically significant pain reduction and pain improvement in function improvement, So we're very excited about these results and couldn't be happier where we are today with the results of the 008 study. But now I'd like to turn our attention to the upcoming catalysts and near-term news flow, which includes the following. By the end of calendar year 2022, we expect the first data safety monitoring board review for Para-OA-002. We expect to commence our extension study for Para-OA-006, and we expect to receive our fiscal year 2022 tax rebate. In the first quarter of 2023, we expect further IP generation and protection to be granted, MPS I data to be presented at the International Conference on Lysosomal Disease, and we intend to share the results of Para-OA-008, the 6-month data that we're very excited to share with you. In Q2 of calendar year 2023. We expect to share data for our Canine OA model at the 26-week mark, which again, as Dr. Krishnan had mentioned, is equivalent to what we would expect to see in humans 3 years post-administration. And we also expect to update you on a stage 1 dose selection for para OA 002. Finally, we continue to engage in discussions with multiple potential partners for our phase 2 asset in mucopolysaccharidosis, MPS. And we certainly will keep our investors informed of the ongoing results of the 6-month data and the 12-month data for 008 and provide continued updates for all of the Paradigm clinical study results. So once again, we thank you for attending today's investor conference call, and we hope that you are as pleased as we are with these top-line readout results. And with that, I'll now turn it back over to Matt.
Thanks, Marco, and also to Donna and Ravi for the overview this morning. That concludes today's Paradigm Biopharmaceuticals investor webinar. Once again, as mentioned at the beginning of the session, any questions today can be referred to the investorrelations@paradigmbiopharma.com email address, and the team will be in contact. Again, given the short notice today for the webinar, there will be a replay available very soon after the conclusion of this, which can be accessed on the Paradigm website and the same link as in the ASX announcement. So thanks again to everyone who joined the session for participating today, and we look forward to seeing you for our next update. Goodbye for now.